Okay, let's unpack this. We all know that feeling, right? You think you've got a handle on something, maybe like the basics of your nervous system, and then you realize, wow, the simple textbook definition, that's just scratching the surface of this huge, complicated setup. Today, we're not just aiming for a quick look, we're really going for that elite professional level of understanding. We're diving deep into the autonomic nervous system, the ANS, and that whole intricate dance of molecules called neurohumoral transmission, NHR.
That's exactly the plan. We're going to connect the dots, from the actual structure of the ANS, the nerves, ganglia, brain bits, right down to the chemical messages flying around every millisecond. These messages control everything your body does automatically. We want to get way past just fight or flight. We're talking ion channels, receptors, actually the surprising mix of chemicals that are all working away beneath your conscious awareness.
Yeah, we've got quite the stack of source material here. It covers everything from, well, T1 down to L3 in the spine, to Loewy's famous frog heart experiment, all the way to how glial cells clean things up. So let's jump in. First things first, let's define the territory. What makes the ANS so different?
Right. So when we talk nervous system, the first big split you got to make is between somatic, that's your voluntary movement, consciously controlled stuff, and autonomic. Autonomic handles, well, pretty much everything else. But the key is they're set up almost completely differently, getting these 6 main distinctions down. That's absolutely essential if you want real mastery here.
Okay, difference number one seems pretty straightforward. Organs supplied. Somatic nerves, they only target skeletal muscles, your biceps, your quads, the ones you choose to move. But the autonomic system, wow, it goes everywhere else. Move the muscle, heart muscle, glands, all your internal organs, the whole internal machine.
And that difference in the target, it changes absolutely everything that follows in terms of how it's wired up. Which brings us to point 2, the distal synapse location. Think about the somatic system. The nerve cell starts in the CNS, travels out, and the very last connection, the synapse releasing acetylcholine onto the muscle. That happens right at the end. The decision point is in the CNS. But the autonomic system, totally different. The connection, the synapse between that first neuron, the preganglionic one, and the second, the postganglionic one, that happens outside the CNS. It happens in these special relay stations called ganglia. This whole setup, these peripheral ganglia. That's classic ANS.
And that architecture then dictates the nerve fiber type, doesn't it? Somatic fibers, they're myelinated the whole way. Super fast signals. Why are autonomic ones different? You said the preganglionic ones are myelinated, okay, but the postganglionic ones, the ones actually touching the organ, they're not myelinated. Isn't that slower? Why do that?
But that trade-off is functional, you see. The signals here, they don't always need that split-second reflex speed. They need to be more modulatory, kind of like adjusting a dimmer switch, not flicking a light on instantly. Being non-myelinated lets these postganglionic fibers be much thinner, spread out more diffusely. It sets them up for that wider, slower, more sustained kind of chemical messaging neurohumoral transmission. It's really built for this pervasive adjustment, not raw speed.
Okay, that makes sense. And this also explains the fourth difference you mentioned, plexus formation. Somatic nerves are mostly direct lines, so no real need for complex networks or plexuses, but the ANS forms these intricate webs, these plexuses, out in the periphery.
Exactly right. Because controlling your insides, it needs coordination, right? Think about digestion. You need signals from different nerve segments working together. These plexuses, they allow this constant mixing and matching, coordination, adjustment over big areas of your internal organs. It's like a distributed control network, always chatting back and forth.
And then the chemistry, primary transmitters. Somatic system is simple, just acetylcholine, ACC, acting on the muscle. But the ANS immediately shows its chemical toolkit. It uses both acetylcholine and noradrenaline, NA, as its main output signals, which right away gives us 2 huge families of drugs to think about.
And that chemistry difference leads to maybe the most profound conceptual difference. This is really key for mastering the concept. The effect of nerve section. What happens if you cut the nerve? Cut a somatic nerve. The muscle it supplies is paralyzed, just stops working. Eventually, it wastes away, it atrophies. Why? Because skeletal muscle needs that nerve signal to even start contracting.
But if you cut an autonomic nerve, the organ, say the heart or the gut, it keeps going, no atrophy. How is that possible?
That's the absolute core difference between initiation and modulation. See, your internal organs, they mostly have their own built-in activity, like little pacemakers or inherent rhythms. The ANS doesn't start the heartbeat. It doesn't initiate the gut movements. It just adjusts them, turns the rate up or down, changes the force, tweaks the secretion. So cut the nerve. You remove the tuning input, sure. The engine keeps running, maybe just not as smoothly tuned, but it runs. That simple fact tells you the whole point of the ANS.
Okay, boundaries established. Now let's map the actual circuit. We need sensors coming in afferents, processors in the middle, the centers, and the movers going out, efferents.
Absolutely. And we have to start with the autonomic efferents. People often focus just on the motor output, the efferents, but these visceral nerves carry signals both ways. They're mixed. These afferents are super important sensory pathways. They're made of non-myelinated visceral afferent fibers.
And where do these sensory nerve cells actually live? Where are their cell bodies?
Good question. Two main spots. Some live in the dorsal root ganglia of the spinal nerves, just like the sensory neurons for touch or pain from your skin. Others are found in the sensory ganglia associated with cranial nerves, like the nodos ganglia, which belongs to the vagus nerve. Functionally, they carry signals for that deep, dull, hard-to-pinpoint visceral pain. And crucially, they mediate vital reflexes adjusting your blood pressure, heart rate, breathing, all automatically based on internal conditions.
Okay, so that sensory info flows up to the command structure, the central autonomic connections. Now, the sources are really clear on this. There aren't any purely autonomic only areas in the brain or spinal cord. It's all integrated with the somatic system. But where's the top boss? The highest level of command for visceral stuff.
That central command post, the highest seat, is the hypothalamus. It's like the master coordinator, balancing our internal state with what we need to do behaviorally. And interestingly, its structure kind of mirrors the ANS's own split. The back and side parts, the posterior and lateral nuclei, they're mainly linked to sympathetic activity. Think stress, energy use, getting ready for action. While the front and middle parts, the anterior and medial nuclei, they lean towards parasympathetic functions. Rest, digest, conserve energy.
So hypothalamus is the strategist. What about the field commanders handling the immediate actions?
Those are the lower centers. Down in the midbrain and the medulla, you've got these dedicated centers running specific autonomic reflexes. Things linked directly to cranial nerves, like controlling your pupil size. The vagus nerve's control over the heart, the basic rhythm of breathing, very specific jobs. And then even lower, in the spinal cord itself, that lateral column of gray matter in the thoracic region, that's where the nerve cells originate. They give rise to the main sympathetic outflow. That's the anatomical root of the dorsal lumbar system.
Right. This whole hierarchical system eventually funnels down to the actual output nerves, the autonomic efferents, the motor side. And as we know, this is where we see that big split anatomically and functionally into the sympathetic and parasympathetic divisions. Let's really dig into those differences now.
Yeah, these two divisions, sympathetic and parasympathetic, you can almost think of them like two different armies. They've got different home bases, different supply lines, different ways they deploy. And that dictates how they respond.
First up, the geography. Origin, where do they come from? Sympathetic is the dorsolalumbar system. Its nerves exit the spinal cord from the thoracic segments T1 down to the lumbar segments L2 or maybe L3. Parasympathetic is craniosacral. It uses specific cranial nerves, third, 7, IX, and the big one, X, the vagus, plus nerves coming out from the sacrospine segments S2 to S4.
And that starting point directly influences their reach, their distribution. Sympathetic has this incredibly wide distribution. Its ganglia, remember those relay stations? They're generally located away from the organs they supply. Often they're lined up right next to the spinal column in what's called the sympathetic chain. This lets a signal spread out very broadly, almost globally. Parasympathetic, though, much more limited distribution, mainly head, neck, and the trunk viscera. And its ganglia are usually located right on the target organ or very, very close by.
Ah, okay. And that location of the ganglia totally flips the script on the fiber ratios and lengths. Sympathetic ganglia are near the spinal cord, so they have short preganglionic fibers coming from the cord, but then long postganglionic fibers traveling out to the distant organ. Parasympathetic with ganglia near the target must have long preganglionic fibers reaching out from the CNS, and then really short postganglionic fibers just making the final jump.
Exactly. But here's the detail that really separates, basic understanding from that elite level, the pre-postganglionic ratio, this number difference. It's the mechanical basis for how they function differently. The sympathetic ratio is huge. It can be anywhere from 1 to 20, up to 1 to 100.
Wow. So one nerve signal coming from the cord can trigger potentially 100 different output fibers. That sounds like a megaphone system.
It absolutely is.
Yeah.
It's engineered for massive divergence, spreading the signal wide and fast. That's what enables that sudden, coordinated, whole-body emergency reaction, the mass action, like flipping one master switch that turns on everything at once. Parasympathetic ratio, tiny in comparison, often just 1 to 1, maybe 1 to 2. This ensures really fine, discrete, localized control, more like a dedicated phone line to one specific device. The only real exception where you see a bit more spread in parasympathetic is within the gut's own nerve network, the enteric plexuses, where you need some local coordination. Hashtag tag tag 2.2 transmitters, stability, and function.
Okay, let's circle back to the chemistry. The choice of chemical messenger really dictates the speed and spread of the signal, right?
Definitely. So the sympathetic system's main postganglionic transmitter is noradrenaline, NA, also called norepinephrine, the parasympathetic system. Its major player is acetylcholine, ACH.
And you mentioned earlier their transmitter stability is quite different. That seems crucial. Na is stable, it hangs around, diffuses a bit, supports wider, longer actions for that stress response, but ACH gets destroyed almost instantly.
By what? By a specific enzyme, acetylcholinesterase, ACE, which is cleverly positioned right there at the synapse, waiting to break it down. This super fast breakdown means parasympathetic actions are quick, they're fleeting, and they stay very local, which is exactly what you want for fine-tuning things like digestion or slowing the heart. You need rapid, precise adjustments, not a lingering global effect.
So the whole system is constantly balancing itself through this, what the source calls the algebra of tone. The activity of any organ getting input from both sides is basically the sum of the sympathetic push and the parasympathetic pull. Functionally, sympathetic gets you ready for stress, mobilizes energy. Parasympathetic handles the housekeeping, digesting food, conserving energy, a constant dynamic tug of war.
Right. And while that check and balance antagonism model is a good starting point, it's not the whole story. Real mastery means knowing where that model breaks down. These two divisions, they aren't always just simple physiological opposites.
Interesting. Where do we see them working together, or at least not directly opposing each other?
A great example is in the heart's atrial fibers. Think about the refractory period. That's the reset time the heart muscle needs before it can beat again. Now, you might expect one system to shorten it and the other to lengthen it. But actually, input from both sympathetic and parasympathetic nerves decreases the refractory period in atrial muscle. They're not fighting over it. They're both sort of tuning the maximum speed the atrium can respond. It's more like synergistic modulation in that case.
Okay, that's subtle. But maybe even more important are the organs that only get single innervation. They're only listening to one side of the conversation.
Yes. And this makes perfect functional sense when you think about it. Organs that get sympathetic, only input. Things like most blood vessels, the spleen, those tiny piloerector muscles that make your hair stand on end, and sweat glands. These are mostly about controlling pressure, temperature, general readiness. You don't really need an active system to dilate most blood vessels. You just relax the sympathetic tone that normally constricts them. A single input is enough to dial the baseline state up or down.
And conversely, some things only get parasympathetic. Only input.
That's right, like the ciliary muscle in your eye that focuses your lens for near vision, or the smooth muscle in your bronchioles, and the main glands in your stomach and pancreas that release digestive juices. This setup ensures really fine, localized control over specific functions like focusing your eyes or releasing specific enzymes without needing a sympathetic countersignal for those particular muscles or glands. Hashtag 2.4, the enteric nervous system, ENS. Now, before we dive deeper into the chemistry of transmission itself, we absolutely have to talk about the enteric nervous system, the ENS. People sometimes call it the gut wall brain, and honestly, that's not far off, given how complex and semi-independent it is.
Right. It's physically separate, embedded right there in the wall of the gut. What's its structure like?
It's basically two main interconnected networks of neurons. There's the submucosal plexus, often called Meissner's plexus, which is more involved in sensing the gut environment and controlling secretion. And then there's the myenteric plexus, or Auerbach's plexus, which sits between the muscle layers and is the main driver of gut motility, the muscle contractions.
And its function is pretty amazing. It gets input from both sympathetic and parasympathetic nerves, sure, but the really cool part is that it can operate independently, right?
Totally. The ENS is the gut's own integration center. It can manage the complex sequences of peristalsis, segmentation contractions, secretion, local blood flow, absorption, all without needing constant moment-to-moment commands from the brain or spinal cord. The sympathetic and parasympathetic inputs, they mainly just modulate the rate or the force of the activity patterns that the ENS is already running. This autonomy is essential for handling the nonstop complex job of digestion.
Our sources also mention its wiring is particularly complex.
Yeah, because the signals don't just travel in one direction. ENS neurons send signals forwards, down the gut, anterograde, but also backwards, retrograde, and even sideways, circularly around the gut wall. This allows for incredibly sophisticated local reflexes. For example, it ensures that when a lump of food is moving down, the gut wall ahead of it relaxes, while the section just behind it contracts to push it along, all coordinated locally by the ENS.
Okay, let's shift gears slightly. Understanding how these nerves talk to their targets. That discovery was a huge leap in physiology. For a long time, people thought it was purely electrical, right? Like a direct current jump. And we now know electrical transmission does happen sometimes in specific places. But the discovery of chemical transmission, NHR, that really changed everything.
It really did. The first clues were based on observation and pharmacology. Way back in 1905, a researcher named Elliott noticed that applying adrenaline mimicked the effects of stimulating sympathetic nerves. He made the conceptual leap. Maybe the nerve itself releases something like adrenaline. Then just a couple of years later, 1907, Dixon saw similar parallels between the vagus nerve, parasympathetic, and a substance called muscarine. He proposed a vagus-released muscarine-like chemical. These were brilliant ideas, but they were correlations, not direct proof.
And the direct proof that came from Otto Loewy in 1921 with that incredible, almost legendary, perfused frog heart experiment, that's a story worth telling.
Oh, absolutely. The story goes he drugged the experiment design, woke up, scribbled notes, supposedly couldn't read them, then dreamt it again and rushed to the lab. It was elegant. He took 2 frog hearts. Heart 1 still had its vagus nerve attached. Heart 2 didn't. He perfused Heart 1 with saline, collected the fluid, and then bathed Heart 2 in that same fluid. So he stimulates the vagus nerve going to Heart 1, as expected. Heart 1 slows right down. That's the parasympathetic effect. But the knockout blow. Heart 2, which had no nerve stimulated, also slowed down and eventually stopped, just from being bathed in the fluid that had flowed through Heart 1.
The only explanation was that stimulating the nerve on Heart 1 released some chemical into the fluid. Loewy called it vagostoff, and that chemical carried the signal to Heart 2. Proof of chemical or humoral transmission.
Yes, exactly. Game-changing. Then came the identification. A guy named Dale had already characterized acetylcholine, ACA, back in 1914, for its parasympathetic mimicking effects. By 1926, Loewy's vagostoff was confirmed to be AC. Identifying the sympathetic transmitter took a bit longer. It wasn't until 1946 that von Euler nailed it down as noradrenaline, NA. And of course, since then, the list of known neurotransmitters has exploded: dopamine, serotonin, GABA, glutamate, peptides, purines. It's the foundation of modern neuropharmacology. Hashtag 3.2. The 4 criteria for a true neurohumoral transmitter.
Okay, so for us aiming for that elite understanding, it's not just knowing the names like ACU and NA. We need the rules. What's the actual scientific checklist? What makes something a true neurohumoral transmitter?
Right. There are 4 classic criteria. These are like the gold standard. First is presence. The chemical, the suspected transmitter, has to actually be found inside the presynaptic nerve ending. And usually you also need to find the enzymes there that the neuron uses to make it. If the neuron can't synthesize it, it probably can't release it functionally.
Makes sense. Criterion 2.
Release. This connects the chemical to the nerve activity. When to stimulate the nerve electrically in a way that mimics natural activity, the substance must be released from the nerve ending into the surrounding fluid or the synaptic cleft. You have to be able to detect it coming out upon stimulation. Third criterion is crucial: identity of action. This is the mimicry test. If you collect the actual substance, purify it, and then apply it directly to the target cell or tissue, the effect it produces must be exactly the same as the effect you get when you stimulate the nerve itself. Identical response.
Okay. Presence, release, identical action. What's the 4th?
The 4th is modification, and it's key for pharmacology. Any other substance, like a drug or another natural compound, that blocks or enhances the effect of nerve stimulation must also block or enhance the effect of the applied transmitter in exactly the same way. This confirms that both the natural nerve signal and the applied chemical are working through the very same mechanism, the same receptors. It validates the pathway for drug development.
Okay, we've got the messengers, we know the rules. Now let's zoom right in on the mechanics. How does the signal actually get across? This whole neurohumoral transmission thing happens in 5 key steps, starting with the electrical pulse running down the nerve fiber.
Right. It all starts with the neuron at rest, the resting state. The inside of the nerve cell membrane is kept at about -70 millivolts compared to the outside. It's polarized. This negative potential is mainly due to the membrane being quite permeable to potassium ions, Tex++ to Waller, which tend to leak out. But crucially, it's maintained by the constant work of the Tex9 plus DOM. This molecular machine actively boots sodium plus out and pulls potassium plus in, keeping those concentration gradients steep.
Then the trigger arrives, and bang, the action potential, AP. This is that sudden, very rapid electrical spike. What's happening there at the membrane level?
The initial stimulus causes voltage-gated sodium channels to snap open. Suddenly, the membrane becomes massively permeable to TXD plus dollar. And because there's way more TXD plus outside than inside, it rushes in, driven by both concentration and electrical gradients. This influx of positive charge causes the rapid depolarization,
Yeah.
flipping the membrane potential so the inside actually becomes positive for a moment, maybe plus 20 milliV. That's the overshoot. Repolarization follows quickly as potassium channels open, letting TXT plus ions rush out, carrying positive charge away and bringing the potential back down. And then that TXT P plus no pump has to work hard during the brief refractory period to tidy up the ion distribution, getting everything back to the resting state, ready for the next signal.
And this AP doesn't just happen in one spot. It travels down the axon. How does it propagate?
The electrical change in one patch of membrane sets up little local currents that flow to the adjacent patch, triggering its sodium channels to open. This wave of depolarization moves along the axon. In myelinated fibers, it's even faster because the current jumps between the gaps in the myelin, the nodes of Ranvier. It propagates without weakening, and because this ionic flow is so fundamental, nature's cooked up some really potent ways to block it. For anyone wanting to master this, knowing about tetrodotoxin, TTX from pufferfish, and saxitoxin, STX from shellfish, is key. What's cool about them pharmacologically is their specificity. They selectively block those voltage-gated sodium channels. No sodium influx, no action potential. They completely shut down nerve impulse conduction. Lethal stuff.
Okay, the electrical AP reaches the end of the line, the presynaptic terminal. Now we need to switch languages. From electrical to chemical, the transmitter molecules are waiting, packaged up in little membrane sacs called synaptic vesicles.
Exactly. And the trigger to release the contents of those vesicles is the process called exocytosis. When the action potential arrives at the terminal, it opens different voltage-gated channels this time, channels specific for calcium ions, TXTA2 plus DEAL. Calcium rushes into the nerve terminal.
Yes, calcium is the real key here, the trigger for release.
Calcium is the absolute molecular linchpin. That influx of TXTA2 does something. It basically makes the vesicle membrane and the nerve terminal membrane more fusible, allows them to dock and merge. Once they fuse, the vesicle opens up to the outside, and everything inside the neurotransmitter, maybe some enzymes stored with it, other proteins gets dumped out into the synaptic cleft, that tiny gap between the nerve and the target cell. This is why things that mess with calcium levels can have such huge effects on nerve function.
But wait, there's an important exception we need to flag. Not all chemical messengers are stored and released this way.
Good point. That's true for mediators like nitric oxide, NO, which is a gas, and things like prostaglandins and endocannabinoids, which are lipid-derived. These guys aren't stored in vesicles. They're typically synthesized on demand, right when the cell is stimulated. And because they're small or lipid-soluble, they just diffuse right across the cell membrane into the cleft. No exocytosis needed.
Now, this release process, it isn't just a one-way street, is it? There's regulation happening right there at the nerve terminal. Presynaptic modulation.
Oh yeah, the nerve ending isn't just dumbly spitting out transmitters. It's covered in its own receptors, called autoreceptors or heteroreceptors, that act like feedback sensors. A built-in dimmer switch or brake. It's quite elegant. Take noradrenaline, NA. When NA is released, some of it can actually bind back onto alpha-10 receptors located right there on the same nerve terminal it just came from. Binding to these alpha-10 receptors triggers a mechanism inside the terminal that inhibits further NA release. So if levels in the cleft get too high, the nerve automatically puts the brakes on its negative feedback. But it's not just NA regulating itself. Other things can inhibit NA release too, acting on different presynaptic receptors, dopamine, adenosine, certain prostaglandins, even opioid peptides like enkephalins can turn down NA release.
So you can inhibit release. Can you also boost it?
Absolutely. There are increased examples too. A drug like isoprenolone, which stimulates data-2 receptors, can actually increase NA release from sympathetic nerves. And significantly, angiotensin, acting via TEX2 receptors found presynaptically, also enhances NA release. Now think about that. Angiotensin is a key player in blood pressure regulation. This means your overall blood pressure status, via angiotensin, is constantly influencing how much NA your sympathetic nerves are releasing moment to moment. A direct link between systemic state and basic nerve function. And notice the detail with acetylcholine regulation. Yes, alpha-2 agonists and muscarinic agonists can inhibit AC release, but primarily at the final junction with the target organ, the neuroeffector site. This inhibition doesn't really happen or happens much less at the autonomic ganglia or at the neuromuscular junction in skeletal muscle. The system seems to protect these critical transmission points from that particular type of feedback inhibition.
Right. The transmitter's been released. It diffuses across that tiny synaptic cleft. Now it bumps into the target cell membrane on the other side, the postjunctional membrane. What happens next?
It binds to specific receptor proteins embedded in that postjunctional membrane. Think of it like a key, the transmitter, fitting into a specific lock, the receptor. This binding event is what triggers the response in the target cell. The response can generally be one of two things, either excitatory or inhibitory.
Okay, let's take excitatory first. That leads to an excitatory postsynaptic potential, or EPSP. What does that mean electrically?
An EPSP means the postjunctional membrane depolarizes, it becomes less negative inside. The resting potential moves closer towards the threshold needed to fire an action potential of its own. The mechanism is usually the receptor opening up ion channels that allow positive ions to flow in. Typically, this involves an increased permeability to cations like sodium, plus to many and plus 2. These positive ions flow in passively down their concentration gradients, making the inside of the cell more positive. There's usually a corresponding efflux of text K plus 2, but the net effect is depolarization.
And the opposite is an inhibitory postsynaptic potential, IPSP. This makes the target cell less likely to fire. How does that work?
An IPSP either stabilizes the membrane potential or actually hyperpolarizes it, making it even more negative inside, moving it further away from the firing threshold. Two main ways this can happen. One mechanism is the receptor increases permeability to negative ions, specifically chloride. Since text of keys is usually more concentrated outside the cell, it flows in, making the inside more negative. The other main mechanism is a selective increase in permeability just to potassium. Potassium is more concentrated inside, so opening text-hexagon chain plus channels lets positive charge flow out, again making the inside more negative or hyperpolarized.
And we shouldn't forget that even when there isn't a strong signal firing, there's often a low level of transmitter release happening. This baseline leak has a trophic influence, right?
Yes, that's an important concept. This slow basal release of transmitter seems to be necessary to maintain the structural integrity and functional sensitivity of the synapse over time. It's like a maintenance signal, keeping the connection healthy and ready. So what's the ultimate outcome of these EPSPs and IPSPs? Step 4 is about the consequence. If you get an EPSP that's strong enough to push the membrane potential past its firing threshold, a suprathreshold EPSP, then the postjunctional cell fires its own propagated action potential. The signal has successfully crossed the gap. If the target cell is another neuron, that means a nerve impulse continues down the line. If it's a muscle cell, it leads to contraction. If the gland cell, it triggers secretion.
And the IPSP outcome, its job is basically to prevent that.
Exactly. An IPSP stabilizes the membrane. It makes it harder for any simultaneous or subsequent EPSPs to reach the threshold. It effectively resists depolarization, acting like a brake or an off signal, ensuring the target cell stays quiet when it's supposed to.
Okay, the signal got across, the target cell responded. Now for the system to work properly, especially at high speeds, that signal needs to be turned off quickly. Step 5, termination. The sources say the speed of termination dictates how fast nerves can fire, maybe up to 1,000 times a second. So how is the transmitter action stopped? Depends on the transmitter, right?
Absolutely. The termination mechanism is tailored to the specific chemical nature of the transmitter. There are basically 3 or 4 main strategies the body uses. Strategy 1 is local degradation. This is the classic method for acetylcholine, ACE. Remember that enzyme, acetylcholinesterase?
Yes.
ACE key. It's anchored right there in the synaptic cleft on the membranes. As soon as ACE binds to its receptor and does its job, ACE chops it up, hydrolyzes it, rendering it inactive almost instantly. This allows for very rapid, brief signaling, perfect for the parasympathetic system's fine control.
Okay, so ACE is broken down. What about noradrenaline, NA, and other similar amine transmitters like dopamine and serotonin? They use a different method.
Yes. The primary mechanism for these monoamines is active reuptake. Strategy 2. Most of the NA release doesn't get broken down in the cleft. Instead, it gets actively pumped back into the presynaptic nerve terminal that released it. This reuptake is done by specific protein machines in the nerve membrane, the transporters. We have NET, the norepinephrine transporter, DD for dopamine, SERT for serotonin. This is incredibly efficient because the transmitter is recycled, repackaged into vesicles, ready to be used again. And crucially, it's a major target for drugs. Think about cocaine or many antidepressants. They work by blocking these transporters, leaving more transmitters in the cleft for longer.
Right, so degradation for LE, reuptake for NA. What about the bigger molecules, the peptide neurotransmitters like VIP or NTY or enkephalins? How are they cleared?
They use strategy 3, which is basically just diffusion and distant breakdown. These peptides are generally too big and complex for specific reuptake transporters, so they simply diffuse away from the narrow synaptic cleft into the surrounding tissue fluid. Once they're further away, they eventually get broken down by enzymes called peptidases, but this happens more slowly and at sites distant from the synapse. This slower clearance contributes to the typically longer-lasting effects of peptide signals compared to ATA or NA.
Okay, and there was a fourth mechanism mentioned, particularly for amino acid transmitters like glutamate and GABA, involving glial cells.
Yes, strategy 4 involves active transport into glial cells. While some glutamate and GABA might be taken back up by neurons, a significant amount is actively transported into surrounding support cells in the brain, the glial cells, specifically astrocytes. These glial cells have their own set of transporters like GAT for GABA, so the glia aren't just passive packing material. They play a really active role in clearing away neurotransmitters from the synapse, helping to regulate signaling. Essential knowledge, that glial involvement.
Okay, for decades, the simple rule taught was basically one neuron releases one type of transmitter, Dale's principle sometimes called. But the sources make it clear. That idea is, well, history. We need to get our heads around co-transmission.
Absolutely. This is one of the biggest shifts in understanding neurobiology in recent times. The reality is, most neurons, both in the periphery and in the brain, release more than one active chemical substance when they fire. They're sending out a chemical cocktail, not just a single shot.
So how does that work mechanically? Are these different chemicals stored together, released together?
Usually, the co-transmitters are stored right alongside the primary transmitter in the same nerve terminal, but often in different types of vesicles. For example, a small molecule like AC might be in small, clear vesicles, while a peptide co-transmitter like VIP might be in larger, dense core vesicles. Though there are exceptions. Our sources note that ATP is often stored in the same vesicles as noradrenaline in sympathetic nerves. Regardless of how they're packaged, when the nerve impulse arrives and triggers exocytosis, both, or all, the transmitters present are typically released together into the synaptic cleft.
Let's nail down the key players here. What co-transmitters do we commonly see paired up in the autonomic nervous system?
Okay, in cholinergic neurons, the ones primarily releasing HC, just common co-transmitters, include VIP, vasoactive intestinal peptide, and sometimes ATP, adenosine triphosphate. In adrenergic neurons, primarily releasing NA, the usual suspects released alongside it are ATC and MPY, neuropeptide Y. And beyond just co-transmission, we now recognize whole classes of nerves defined by other transmitters, like nitrogen nerves, especially at some parasympathetic sites where nitric oxide, NO, acts as a key transmitter, either alone or perhaps alongside ATP. Hashtag 5.2 functional roles of co-transmitters.
So why the extra complexity? Why send out 2 or 3 signals when it seemed like one worked okay for a long time? What's the point of co-transmission?
It allows for much more sophisticated signaling, more nuance, more control, especially over time. One major role for co-transmitters is neuromodulation. They can act back on the presynaptic terminal to influence the release of the primary transmitter, maybe fine-tuning it, making it stronger or weaker on the next pulse. Or they can act on the postsynaptic cell to change its sensitivity to the primary transmitter, like adjusting the volume knob on the target cell's receptors, so they're tuning the main communication line.
But they aren't just modulators, right? They can have their own direct effects, too.
Oh, definitely. They can serve as an alternative transmitter in their own right. NPY, for instance, which is released with NA from sympathetic nerves, causes very potent and long-lasting constriction of blood vessels. That's a direct, powerful effect, independent of NA, though often complementary. And just like the primary transmitters, these co-transmitters can also exert that background trophic influence, helping to maintain the long-term health and structure of the synapse. Hashtag 5.3 time course and diffusion differences. One of the really clever things about co-transmission is that the different messengers often have different time courses of action. They don't all hit the target and wear off at the same speed. This lets a single nerve firing produce a complex response that unfolds over time.
Can you give some examples from the ANS? Let's take parasympathetic first.
Okay, so imagine a parasympathetic nerve firing that releases ATH, NO, and VIP. The ACH gives you the very fast, immediate effect, maybe a quick muscle twitch or gland secretion, lasts milliseconds. The NO, being a diffusible gas, might have a slightly slower onset but perhaps a bit more sustained action, maybe lasting seconds perhaps, causing local blood vessel dilation. And then the VIP, the peptide, takes longer to act because peptides often trigger slower intracellular signaling cascades. But its effects can be much more slow and long-lasting, maybe minutes, ensuring the overall response lingers appropriately. You get this multiphasic response from one nerve signal.
And we see a similar pattern on the sympathetic side with NA, ATP, and PY.
Exactly the same principle. When a sympathetic nerve fires releasing all three, the ATP often produces the fastest effect, maybe initiating a quick spike in muscle tension via its own P2 receptors, faster even than NA. Then the NA comes in, providing the main, more sustained phase of the response, acting through adrenergic receptors. And finally, the NPY, being a peptide, has the slowest onset but the most prolonged action, contributing to really long-lasting effects like sustained vasoconstriction or changes in tissue growth, fast, medium, and slow components from a single event.
And you mentioned peptides and NO diffuse more widely, too, because they're not rapidly taken back up or broken down right at the synapse, so they have a wider influence.
That's right. Unlike AC or NA, whose action is mostly confined to the immediate synaptic cleft, co-transmitters like NO, VIP, and NPY can diffuse further away, potentially reaching receptors on nearby cells that weren't directly innervated. This gives the system a way to achieve a broader, more regional influence that goes beyond just the direct point-to-point wiring. Hashtag 5.4 non-adrenergic, non-cholinergic NANC transmission.
This whole idea of multiple transmitters leads us straight to the concept that really cemented the complexity: non-adrenergic, non-cholinergic transmission, or NANC. This was needed to explain responses in organs, especially the gut, that just didn't fit the old A-Chenet model.
Precisely. NANC transmission is defined experimentally. You take a tissue, maybe from the gut, the bladder, the vas deferens, certain glands. You stimulate its nerves, and you observe a response contraction, relaxation, secretion. But critically, this response persists even when you have pharmacologically blocked all possible effects of both acetylcholine, using cholinergic blockers like atropine, and noradrenaline, using adrenergic blockers like phentolamine and propranolol. If the nerve stimulation still causes an effect after you've completely shut down both classical pathways, then the transmission must be mediated by something else. That something else is NANC transmission.
Let's use that concrete example from the source material, the guinea pig vas deferens. How does that illustrate NANC and co-transmission?
It's a classic preparation. When you stimulate the sympathetic nerves supplying the vas deferens muscle, you don't just get one simple contraction, you get a biphasic response. Two parts. There's an initial, very rapid, short-lived twitch, and then there's a second, slower to develop, but much longer-lasting phase of contraction. Through careful pharmacological dissection, researchers figured out what caused each phase. The initial fast twitch. That's mediated by ATP, released as a co-transmitter with NA, acting on P2 receptors on the muscle.
Okay, so ATP causes the first quick phase. What about the second longer phase?
That second, more sustained contraction is mediated by the classical sympathetic transmitter, noradrenaline, NA, acting on alpha-1 receptors. So if you block the P2 receptors, the ATP receptors, you lose the fast twitch, but you still get the slow NA contraction. If you block the alpha-1 receptors, the NA receptors, you lose the slow contraction, but you still get the initial ATP twitch. It perfectly shows two transmitters released together from the same nerves, working on different receptors with different time courses to create a complex, functionally relevant response. That's co-transmission in action, and ATP here is acting as a NANC transmitter relative to the classical view.
Wow. Okay, so what does this all really mean then? We started this deep dive talking about just involuntary control, but we've ended up tracing things from brain centers right down to specific ion channels and receptor subtypes. We've gone from the basic T1L3 map to seeing how visceral control is this incredibly dynamic conversation using multiple chemical messengers, feedback loops, even a whole separate nervous system in the gut. It's way beyond a simple on-off switch.
It really is. And connecting it back to that goal of mastery, it means seeing why the system is built this way. The sympathetic structure, long postganglionic fibers, that huge 1.110 divergence ratio is perfectly designed for a rapid, global emergency broadcast, while the parasympathetic structure's short postganglionic fibers, rapid A-sheet breakdown, that tight 1.1 ratio is ideal for precise, localized, moment-to-moment fine-tuning. Understanding this structure-function link is key to predicting drug effects. For me, two of the most striking takeaways are first, just how fundamental that difference is between ANS modulation versus somatic initiation. Visceral organs have their own lives. And second, the sheer reality of co-transmission, throwing out the one neuron, one transmitter rule and embracing this multi-messenger complexity using peptides, purines, gases alongside the classics. It changes everything. It does. And it leaves us with a really interesting, maybe slightly unsettling final thought. We've seen that the release of transmitters like NA is modulated by things like prostaglandins and angiotensin acting on those presynaptic receptors. Prostaglandins are heavily involved in inflammation. Angiotensin is central to blood pressure. So it raises this provocative question. How much is our fundamental day-to-day autonomic balance, our heart rate variability, our gut function, our vascular tone being constantly tweaked, maybe even subtly disrupted by underlying factors like chronic low-grade inflammation or fluctuating blood pressure, precisely because these factors influence the release of the very chemicals controlling those organs? Something to definitely mull over until our next deep dive.